Now, in the last part, we discussed about the transcription and how to synthesize and process your messenger RNA. Now, this messenger RNA carries the information for building proteins to ribosomes and the tRNA for translation. Now, protein building information is carried in what you call the codons. So, these are the sequences of 3 mRNA nucleotides encoding for a specific amino acid. So, basically, remember, we have 4 bases. So, basically, it's an information encoded in 4 characters. And then we have your protein. The protein is a polymer also, but it uses 20 characters instead of 4. So, it's not a 1:1 translation. Now, A is equal to this one, B is equal to this one. It's not. So, in order to accommodate, for example, you have 4 versus 20. So, the bases, the RNA bases, the nucleotide bases are grouped into basically groups of 3. So, those groups of 3 are called the codons. So, these are sequences of 3 and 3 nucleotides. So, they do not overlap. So, there's no overlapping of a codon. So, 3, for example, the first, in a codon, in the start of a G, the first 3 is codon 1, and that's nucleotide 1, 2, and 3. And then nucleotide 4, 5, and 6, that's codon number 2. And then 7, 8, and 9, that's codon 3, so on and so forth, until the end of the gene. Now, each codon codes for specific amino acids. Now, the order of this codon dictates the order of the amino acids in your polypeptide chain. Now, remember, we have 64 codons. So, why 64 codons? Because, again, we have 4, basically a permutation of 4 grouped into 3. So, but the order is also important. So, it's a permutation. So, you have 4 times 4 times 4 times 4. So, you have 64 possible combinations, or not combinations, 64 possible codons. Or basically sequences of 3 among the 4 nucleotides. So, we have 64 codons and we have 20 amino acids. So, basically, there are some amino acids that are coded by more than one codon. So, we have redundancy in such that, for example, some amino acids are encoded by 3 or more codons. So, for example, this codon codes for alanine. And then another codon, still alanine is the one or rather, alanine is also the one that corresponds to that codon. And then we have special codons that signals the start and the end of the gene. So, the translation always starts with the AUG, which is the codon for methionine. So, it's called the start codon. And then you have the stop codons. You have 3 stop codons, and these codons do not encode for an amino acid. So, these stop codons instead signals that the translation should stop, and basically the translation complex should disassemble. So, basically, that's the stop codon: UAA, UAG, and UGA. Now, this is your genetic code. So, the genetic code is basically the cheat sheet of biochemists. So, this one, or not only the biochemist, but also the cell. So, this is basically the list of the codons you have and what type of amino acid corresponds to that. So, here, for example, let's say the start codon. The start codon is AUG. So, you have the first base is A. So, look at the left, on the left side. So, this is the first base, and then the top indicates the second base. And the one on the right is the third base. So, the first base, you have AUG. So, A, and then U. So, you have A, U, and then the G is here. So, A, and then this is the column for U, and then this is the row for G on the third side. So, you have AUG. And this encodes for methionine. Now, let's see another codon. So, let's say CUC. So, CUC, let's look for C. So, C, the groups of C's in this column. So, C, and then U. So, this is U. And then look for C. CUC, so that encodes for leucine. So what else? AAG, for instance. See, you have your A, and then another A. So this is the A column, A, and then look for G. AAG, this is lysine. So another amino acid, lysine. So as you can see here, so you have several codons encoding for the same amino acid. For example, this one, threonine. So 4 bases, or rather 4 codons encode for threonine: ACU, ACC, ACA, and ACG. As you can see, usually it's the third bases that all, even though it varies in the third bases, it's the first and the second that remains consistent for threonine. So they call it the wobble in your, well, basically in biochemistry. So that means that even if there's a mutation on the third codon, it's relatively okay because it still encodes for the same amino acid. We will talk about mutations in a while. But anyway, that is how do you read the genetic code. So if you read it, for example, how about the stop codon? For the stop codon, it's UAA. So the first base is U, and then you go to the A, and then another A. So this is the stop codon. So these three are the stop: UAA, UAG, and UGA. So those are stop codons in your basically genetic code. So looking at the genetic code, you can know na what amino acid should go on that specific codon or position. Now let's move on to the actual translation. So here is your information from the gene in the DNA, and then you transcribe it to your messenger RNA. Now the messenger RNA, you will be dividing them into codons. So the first 3, that's the start codon, and then the next codon. So in here, you need to look for AUG first, and then count off. So once, because this is not the actual end starting end of the transcript, but rather there are more bases here. But it's the start of the translation because it has the AUG. So basically, the start of the translation is where AUG is in the transcript, or rather where the first AUG is in the transcript. So you have AUG, and then you now separate them into your codons, into triplicates. So you have, or rather triplets. So you have AUG, that's methionine. UAC. If you look at UAC, so this is U, A, and C. A, B, C, UAC. That's tyrosine, UAC. So this is tyrosine. And then UCA, that encodes for serine. GAC, that encodes for aspartate. Now, you have your, remember, there is no overlap of the codons. So the codons are adjacent to one another. So you read them basically just like how you read words in a sentence. So basically that's that. So that's your translation. Now let's move on to the different types of the RNA that is involved in the translation step. So you have the transfer RNA or the tRNA that delivers the amino acid to ribosomes. So it contains an anticodon complementary to an mRNA codon. So it contains a binding site for the amino acid coded by your specific codon. So you basically have what you call an anticodon. So the RNA, aside from the tRNA, you have the rRNA. rRNA plus additional proteins, they make up your ribosome. So the ribosomes is the one that actually synthesizes your polypeptide chain. So it links amino acids into the polypeptide chain. So you have 2 rRNA subunits in proteins that make up each ribosome. So here is the ribosome. So you have two different subunits. Basically 2 parts. It's just like say a burger bun. So you have a top bun and a bottom bun. So we have a larger top bun, which is your large subunit, and a smaller bottom bun, which is your small subunit. So you put them together, you form the working ribosome. Now in the ribosome, your messenger RNA is basically sandwiched between the large and the small subunit of the ribosome. And then this is your tRNA. The tRNA is your, well, it's somewhat like T-shape, so it's the one that delivers your amino acid. So this is the RNA part, and this is the amino acid. Amino acid is actually smaller compared to the polymeric tRNA. So here is your amino acid, and this is the anticodon. So the anticodon is the complement of the codon. So for example, this is the start codon. or rather this is the tRNA that delivers methionine. Methionine is the start of your transcription. So the start codon is AUG. Now, as the tRNA that delivers the methionine, it has the complement of AUG. So the complement of A is U. Remember, this is still RNA. So instead of T, you are using U. So A to U, or AUG. So next is U. So the complement is A. And then the last base is G. So the complement of that is C. So the anticodon that this tRNA carries is the UAC anticodon, which complements your AUG start codon. So this particular tRNA carries methionine. Because if you remember the genetic code, AUG corresponds to methionine. So here is your methionine. So you add them, or rather, it's basically how your cells actually translate it. So you add it in the, or rather, the anticodon will pair with the codon in the messenger RNA, and then the amino acid it carries will be added to the nascent or the growing polypeptide chain. So the translation converts genetic information from your mRNA to a new polypeptide chain. So the amino acid order is determined by codon order. So this translation step for eukaryotes, it occurs in the cytoplasm, and we have 3 stages: initiation, elongation, and termination. Now, during the initiation step, you form the complex. So first, the smaller ribosomal subunit, the bottom bun of the burger bun ribosome, binds to the messenger RNA. So after that, you have your initiator tRNA. We call it initiator because it's the start. So the initiator tRNA looks for the AUG in the messenger RNA. So its messenger RNA is large, it's actually long molecule. It contains a 5' cap, but it will search for the AUG codon. And then after finding the AUG codon, the tRNA of course base pairs because it contains a complement, the anticodon, so it base pairs here. And then once it pairs up or hybridizes, the large ribosomal subunit, which is the top bun, joins the small ribosomal subunit. Now, once they are joined, so basically that's your initiator complex. You have your ribosome, the two halves, small and large subunit, sandwiching your messenger RNA, and the initiator tRNA is in the inside, or rather is attached, or it forms it forms hydrogen bonds with its complement in your messenger RNA. So you have you have the RNA, the ribosome, and your messenger RNA to form basically the complex. Now, during the elongation steps, your ribosome then will assemble the polypeptide chain as it moves along the mRNA. So the first initiator tRNA is contains methionine, and then after that, the second tRNA will come. It will enter there's a notch in the large ribosomal subunit for the second ribosome to enter. So it's usually called the entry site. So you have the next the next tRNA that encodes for the next codon on the line. So AG, the codon after AUG. So the one that corresponds the tRNA that contains the anticodon for that will enter, and that tRNA carries the amino acid. So your the next amino acid will be joined by the ribosome to form your nascent polypeptide chain. So it will form, the methionine will bond to the next amino acid to form your peptide bond. And then the tRNA will exit. The tRNA will let go of the methionine. It will exit your ribosome, and then the ribosome will move ahead. So the next, ready for the next codon. So when it reaches the stop codon, the ribosome, when the ribosome encounters the stop codon, it will, instead of a tRNA, the release factor will bind. And then when it binds, you will have an enzyme, or rather, the entire complex, the translation complex, will be basically disassembled. So the enzymes will detach the mRNA and polypeptide chain from the ribosome, and that ends your translation. So here is a better view of gene expression basically. So here is transcription. The first step is transcription. So you are copying the information from the DNA using the RNA transcript, and then the RNA is transported outside the cell. So this is your messenger RNA, and then you have the formation of this is basically the translation step from 3 onwards. So you have your initiation complex formation. So the ribosome will form with the tRNA. This is the initiator tRNA. And then you do elongation. So a new chain forms. So basically, from a single mRNA, you can have multiple ribosomes working on it. So as you can see here, this is the growing chain. This is the tRNA that is done its job. So it exits the ribosome. And then this is the next tRNA entering the ribosome. Basically, it contains the anticodon complement of the codon, the next codon in the line. So it carries your amino acid here. And so after doing that, it will join. It will form the amino acid will form a peptide bond and then again exit. Okay, so after that it will exit. So the polypeptide chain will grow longer and longer until it terminates. So this is the translation step. So a more detailed view of that. So this is the start. So you start with the initiator. Start codon. So you have AUG for the stop codon. The initiator tRNA contains the anticodon for that, UAC. It has methionine attached. That's the first amino acid in the polypeptide chain. That's always the first. And then the next codon is GUG. So its complement is CAC that contains valine. So this one will hybridize to this part. So again, base pair. Now, when it base pairs, a peptide bond forms between methionine and valine. And then after that, the first codon, the previous one, will detach itself. So it will exit the ribosome, but it will leave the amino acid behind. That's part of the growing amino acid chain. And then your ribosome moves. It moves forward, three bases forward. So that's another codon here. So that UUA, the anticodon is AAU. So the one that contains that is this tRNA carrying leucine. So again, the process continues. So it will attach here and then it will form a peptide bond. So you will have a growing amino acid chain. So until you reach the stop codon. So this is the stop codon UAA. So when it encounters the stop codon, the complex will disassemble. So basically that is your translation step. Now, in the next video, we'll be discussing about mutations and what are its consequences.